Now, what happens? See, every time I share that, progress, where'd it go? Now it's gone all together. Gosh. All right, I think I'm back. Holy cow. Technical difficulties. All right, if you are zooming and not seeing the PowerPoint, give me a quick shout out. Should be in action. So we left off Friday. I think this was my last slide. I feel like this was the last one. Talking about all of the different classes of immunoglobulins. Handfuls of these would be the participants that we were really just looking at with that immune response as those B cells start pumping out antibody. IgM followed by IgG are the immunoglobulins that are secreted when we think about combating these invaders. There are other players like in the mucus, the IgA, on the surface of cells, the of B cells, IgD molecules that are allowing them to recognize these invaders as well. And important when we think about memory cells. And then with allergic reactions, the IgE, kind of when our immune response goes a little bit haywire. And there are some pretty interesting details with some of this. We will touch a little bit on that later when we think about our immunity doing stuff that maybe we don't want it to do. So one of the mechanisms that some of these antibodies can do, the IgG and IgM, although we're really just looking at G up here, is neutralization. So sticking to something and basically taking it out of commission, making it so that it can't function, neutralizing virus, linking up to spike proteins, different toxins can be stimulated, stimulate an immune response, and antibodies can link up to those toxins, barring their entry into cells like diphtheria or other components of the cell, making it unable to move, potentially neutralizing. So one mechanism. The other, as those opsonizing agents, the little tags on these microbes, when the antigen binding region of that antibody links up to that invader, whoever it may be, maybe a bacterium right here, the FC, the tail region of that antibody, is poking on the outside, and that allows specific cells to recognize that. One of them being a macrophage who has receptors on the surface, FC receptors. So in this instance, the antibody is specific to that respective fragment on that pathogen, the epitope, but the FC region is common. So the FC receptors, we're not looking for specific things, we're just looking for antibody. And if we see that, we will engulf it and pull it in and destroy. So they opsonize to coat it, stimulating the macrophage to ingest it, enhancing some of those phagocytic capabilities of the macrophages that we've already gone through the process of antigen presentation, showing them to T cells, who show it to B cells, who start pumping out antibody, and now it stimulates other macrophages to mop it up a bit quicker, if you will. So another good potential with these antibodies is agglutination reactions. And if we're talking, I mentioned this before, if we're talking red blood cells, we would call it hemagglutination, but it's the same idea, just clumping up. So antibodies recognizing those specific epitopes and linking up, tethering multiple cells, whatever they may be, together, which in turn sets the stage for them to be taken out of commission, if you will, but also slowing them so that other immune cells have the capability of coming in and destroying. Not only are we thinking cells like macrophages and neutrophils that have capabilities of this, but also those complement proteins that get stimulated by IgG and IgM. Again, that the tail, the FC region, stimulates those complement proteins. And so that helps once they're all bound together with all of the players, proteins, and cells combined, it results in the destruction of these respective cells, whoever they are, bacteria in this example. So the next bit we see with our IgG, and this is the antibody-dependent cell-mediated cytotoxicity. So we need to have those antibodies, all right? And in this case, we talked natural killer cells before, and natural killer cells can function on their own without recognizing antibody. And they can see different things like maybe virally infected cells. They're looking at our own cells, at those MHC molecules on our surface, the MHC ones that say, hey, this is me, all right? And if they recognize that that's not me, because my cell isn't producing that protein like it should, those natural killer cells get stimulated and cause those cells to die. But they also have FC receptors on their surface, like the macrophages, that allow them to be stimulated by antibody as well, giving them multiple ways to do their job. So when they come in contact with something that's been opsonized, that's been coated with antibody, it stimulates the destruction of those cells. So the natural killer links up to the FC region and it extracellularly kills. It starts dumping digestive enzymes onto the surface of these cells and destroys them, these cytotoxins. So multiple ways, and there are numerous cells that have multiple ways to recognize and combat. So that overall for our body is a leg up. It's an advantage that they've got multiple potentials to recognize and destroy. So this image shows the MHC1s and MHC2s, all right? And with that, MHC1 molecules, and these are multiple subunits, each with the letter and number designation. So 4 different polypeptides coming together to give you the functional molecule, the quaternary structure of that protein. The MHC1s are found on all of our cells, all of our nucleated cells, all right? All of our normal body cells, except for red blood cells, harbor these proteins. And through surveillance, stuff like natural killer cells are checking these and making sure, yep, that's mine, all right? And I mentioned before, this is important when we think about organ transplants, trying to get a match as close as possible to this protein structure so that there's minimal rejection, all right? But unless we're thinking identical twin, probably aren't going to have a perfect match. So there's issues with that, all right? So what we're looking at here is really more like a macrophage because we have class 1 and class 2 major histocompatibility complexes. The two together are found on macrophages, the dendritic cells, and B cells. And this allows for the communication between these cells, all right? Recognizing and really thinking, the show and tell sort of stuff. Those epitopes that were on the surface of those pathogens, whatever they are, when the pathogen gets destroyed and they get processed, the epitopes, as these protein subunits are coming together in Golgi, getting packaged all up, those epitopes also show up, and we package that epitope right up here in this cleft, this antigen binding cleft. That's where the epitope gets displayed, and it gets packaged inside of that cell and exported or brought to the plasma membrane, where it anchors, tethers itself in the membrane, and now studded on the outside are a plethora of these epitopes, all sorts of different antigens. And oftentimes, when we think about a response like this and looking at antibody, there's multiple antibodies that have been generated. We call it a polyclonal response, many types. There's research where we truly make one thing, and there's specialized mechanisms of dealing with this. Lab animals get employed for the production of this, but only making one. And we call those monoclonal antibodies. And oftentimes, like in immunodiagnostic assays, monoclonal antibodies get employed because now we're focused in on just one specific thing. But deep down in an immune response, it's not just one antibody, all right? This polyclonal response gives you the potential to recognize all sorts of stuff. And it's all presented right here, all right? So these cells are the show and tell cells, all right, that have the ability to show those epitopes. They also have this, all right? So all nucleated cells, or white blood cells included, have those. So the video showed these dendritic cells going around, sampling everything, destroying and presenting, and really that's the idea. And you can see basically what I just described, these MHC2 molecules and the epitopes getting packaged together and exported to the surface of these cells, now ready to start the show and tell process to stimulate all sorts of other activities to hopefully combat whoever it is that's made its way in. So all of these cells have numerous types of protein molecules studded on their surfaces that lend to the ability of communication with other cells, recognition of other molecules. And on the T cell, here's our plasma membrane, there is a protein region or a protein that's protruding through there, not the same as our MHC2, but a similar idea. These are going to be recognizing the MHC2s on the surface of those other players for the opportunity of some exchange, if you will. All right. So not the same as an antibody, but it's similar. There is a constant region, like the tail region of that immunoglobulin. It's tethered together through some covalent bonding, disulfide bond, linking the alpha and beta subunits of these proteins together. And up at the top, like the Y of your antibody, is this variable region. And just as the video indicated, there's dendritic cells showing these things, but they need to bump into the proper cell that's got the proper assortment within this variable region, this antigen binding site, that allows it to recognize this is the piece that I've been looking for, that missing jigsaw puzzle piece, right? This is the piece. And once they make contact and recognize the pieces fit, a cascade of events is going to follow after this, all right? So that's the idea with our T cells, T helper cells that are making these recognitions. So that's the idea with this. Our dendritic cell that has shown this little bit. This is a blow up of the dendritic cell, and our little green guy right here is our T helper. All right. So as we showed before with the classes of cells, and all originating from that hematopoietic stem cell and receiving specific cues to differentiate, the T cells start off as they've already differentiated the littlest bit, and we're a naive helper T cell. There's other types of T cells. So we've already differentiated into the helper line, but there's different types of helper T cells. And so this first bit with what's being shown is going to help this T cell decide who he's going to be when he grows up. All right, is basically what's up. So this activation here, the naive cell that hasn't yet decided who am I going to be, helper one, helper two, memory cell technically is going to result from these being stimulated. All right. It's not just going to be like, I'm just going to be a memory cell. All right. This happens first. All right. So T helper ones and T helper twos are what ultimately gets stimulated based off of this fragment that's being shown. All right. So with that said, there could be another epitope on one of these other arms that shows a different naive helper T cell. And so we could have an opportunity where some of these naive cells are stimulated and differentiate into the helper ones, and others on a different arm are stimulated and differentiate into the helper twos. It's important to be able to recognize from multiple arms. All right. T helper ones are important with cell-mediated types of reactions, bringing in or helping neutrophils and helping macrophages to do their job of killing. T helper twos are the ones that do the talking to the B cells and stimulate that antibody production within those B cells. T helper two, humoral immunity. T helper one, cell-mediated immunity. All right. All dependent upon that puzzle piece, that epitope that's being shown. All right. And once they've been stimulated, that's when we think about generation of some memory cells. All right. So memory cells with both helper ones and helper twos. All right. As they start to proliferate. So another line, not the T helper. All right. This is a cytotoxic T cell. All right. And the T helpers, I don't know if it showed it on the previous slide. I'll back it up. Oh, it does. The T helpers, we call these cells CD4 cells. And it's the specific, we'll get a blow up of this in a second, but part of this communication molecule on the surface, this protein, CD4 protein, all right, helping to dock to that MHC2 protein and recognize right thing, all right? So CD4s, helper ones, helper twos, all right? The CD8 cells, it's a different protein, all right? They're different. And so at this differentiation to decide with this T cell, we can have naive helpers or naive cytotoxic ones. They're different right as they've started to differentiate, and then we get activated. All right. So here we've got the cell, and in this case, it doesn't look like our dendritic cell. This is more like a macrophage. Doesn't matter. All right. Either one of these could be doing this talking. All right. So it's showing its epitope, but in this instance, it's bumped into a cytotoxic that's got the proper receptor to see this epitope. They dock, and then they basically exchange. This epitope is going to be moved from the MHC2 on our phagocytic cell over to that cytotoxic cell or the T helper cell. All right, either one of them is going to pick that up. So in this instance here, with our cytotoxic T cell, it recognizes and destroys. And in this case, maybe, oh, this says MHC1, not our MHC2. So this is our own cell, our normal cell. I misspoke with that. Our own body cell showing something on there that's wrong. All right. That in turn stimulates it for destruction. And it releases all sorts of digestive enzymes, similar fashion, like those natural killer cells. They're not the same, but similar sort of function for the destruction of those cells. Cell suicide, apoptosis, all right, is the idea with that. So in this instance, back to the macrophage with the MHC2. So there are numerous things that can potentially happen in these circumstances that maybe don't happen exactly like we'd like it to happen within our body. All right. There are some things that can potentially go wrong. All right. So the idea with this, our receptor, our epitope, so it's trying to recognize, but the piece of the puzzle isn't fitting. All right. It's not recognized. So shape wise, right? It doesn't fit. All right. So in this instance, we don't activate this T cell because we're not recognizing it like we should. All right. It doesn't get stimulated. But there are circumstances where we can do that and make them link up without this recognition. So these super antigens, substances that are being released by these microbes, and it seems a little counterintuitive that this would occur, but it sets the stage for bigger problems. All right. So the super antigen doesn't get recognized by these receptors, but it bridges the gap and it allows them to connect, even though, as you look here, there isn't even an epitope in there. All right. So these cells get stimulated without a stimuli. All right. The super antigen forces this, and it causes these T cells, it's non-specific, uncontrolled activation, and they start spewing out cytokines. We call this a cytokine storm. All right. Bad news when we think about the immune response. All of these chemical messengers are now just getting flooded out, stimulating all sorts of activity, increased inflammation, lots of cells coming in and trying to respond. And truly, what's happening when this bridge has been gapped with these super antigens is that pathogen making an attempt to overwhelm the immune response, make it work itself to death. You can't do anything if you're just spewing this stuff out. You can't communicate with anybody else, and all of these other cells trying to show up, it overwhelms our body, and it also starts to overwhelm all of the cells trying to deal with it. So it results in excessive inflammation, potentially fatal consequences in a cytokine storm. Definitely issues. So one way that pathogens have come up with strategies to look at the immune response and be like, oh, what would happen if we just turned you on full blast? Can you cope with that? Can they cope with that? Could be, it may be a dead end for both. All right. So back to the B cell. This is a membrane on the B cell and those antibodies protruding to the outside. What ones are these? Oh man. IgD studs the surface of those B cells, right? So with that same epitope, like the IgG or the IgM that these cells are spewing out, they also generate some IgD that they stud on the surface of their cells. Same sort of fingerprint within this antigen binding site. All right. We're spewing out the antibodies to link up to stuff. We produce antibodies to stick onto our cells. And as we differentiate into plasma or to memory cells, we still have that recognition molecule on the surface. So subsequent infection, the B cells already got it on the outside and it bumps into that epitope. And it's like, I saw you before. Now I'm spewing out mass quantities of antibody, all right? Giving us some protective immunity. So this next bit, this is T cell independent B cell activation, all right? So we have these B cells with those respective antibodies on their surface. These guys have already been primed previously, all right, to have these immunoglobulins on their surface, all right? But now we, may think second infection or something like that. We don't need that T cell to stimulate. It's already primed. And so we can recognize those respective epitopes and spur a reaction, all right? And start, in this case, recognizing, and now plasma cell pumping out those antibodies, all right? The IgM, and in a little bit, we'll start thinking IgG, all right? Typically, and I told you as we showed that image with the primary and secondary response, there are multiple players and they don't all get spewed out simultaneously. So usually the first bit of immune response, the antibodies are IgM. And shortly after IgM is being produced and secreted out, IgG is the next molecule, all right, next antibody. Again, same variable region in those antibodies. If we're talking the same B cell that's been primed, all right, spewing out antibodies that recognize, all right, and the potential for B cells to shift from the production of this type of antibody to another type of antibody provides opportunities for numerous immunoglobulins to be secreted and stimulated, all right? It also sets the stage for potential issues like, wait a minute, let's secrete some IgE instead, right? And now allergic reactions are a result as we potentially transition, all right? So now this is more really the video showing this activity. The T cell dependent activation of B cells. Here we've got this B cell recognizing, all right? It, with its receptors, it has MHC2s as well. The B cells were one of those players. So in this process, not only are we thinking about stimulating antibodies to be produced just from this initial activity, independent, we also can activate cells. Why not try to stimulate all of it, all right? So the B cell, once it starts doing its job, will present some of those fragments on its surface. We're still thinking about trying to make antibody, all right? But we want to tell T cells, call in more troops. So we show those fragments and those T cell helper cells start communicating with other B cells, and we see more proliferation of this, all right? With that clonal expansion, generating memory, and boatloads of plasma cells secreting numerous immunoglobulins, massive amounts being flooded out of these cells. So this, as I said, is going to be broken down to show you some of these details. So we saw it before with them really merged together, all right, with IgM and IgG both included in that initial figure. This one breaks them apart and shows us, gives us a little bit more detail, all right? So the initial exposure at day 0, taken almost 2 weeks, about 2 weeks, to start seeing a measured response. And that first bit of antibody that is detectable at about 2 weeks really is IgM, all right? And at about 3 weeks into that infection is where we see a peak of our IgG. So just as M is starting to kind of decline, as it's backing off in its quantity in the serum, IgG is starting the uptick and it's starting to take over, and it's got considerably more levels, higher concentration in the serum than IgM, all right? A little more pronounced, all right? And then it starts to decline. And we got our little jig jag in here because the time frame can be varied, all right? Maybe it's a couple of years, or maybe it's only a few months. Who knows how long it's been from this first stimuli to the next, all right? The time frame is varied after this little bit. So some time passes, we get a secondary infection or another exposure to it, or a booster shot if we're talking vaccination, doesn't matter, all right? That second exposure to the antigen, IgM, now we have shifted it about a week instead of 2. We start to see its level increasing, and long before we really started the decline, IgG is being produced as well. And it surges past. Both levels of IgM, basically about the same. Primary to secondary response, you don't see a measured increase in IgM, but you definitely do with IgG. And really the result of that are those plasma cells that have converted into memory cells that were IgG. There's a lot more of the memory cells that are G than M that stimulates this rapid increase, and it stays prolonged, considerably greater than our initial primary exposure, all right? And at this point, we think about the potentials really for some immunity, all right? Not every type of infectious agent confers immunity, like lifelong immunity, but many we think about truly do, all right? You've probably heard stuff like, the common cold. There are hundreds of different variants, strains of cold viruses, numerous types of viruses that cause the common cold. You get infected with a cold virus, you combat it, and you produce antibodies against it. You don't get the same cold virus and mount an infection against that. You get a different cold virus, but the signs and symptoms are basically exactly the same, all right? A cold's a cold, right? So every cold, you've produced antibodies, all right? You've produced some protection. And as you get older, you have produced boatloads of different antibodies against a plethora of different cold viruses. And deep down, I think that's why grandparents are such good babysitters. When a kid gets sick and can't go to daycare or to school, but you still have to go to work, dumping them off at grandparents' house might be okay, right? And you might be like, wait a minute, you don't want to get grandma sick. Grandma's probably not going to get sick because she probably had that cold 35 years ago, all right? And has immunity against that specific cold. Maybe she gets sick, all right? But likely not, all right? I think that's impressive. And I don't know, so many, all right? So IgG and IgM, classical type of antibody response against some pathogen, shows this type of activity. And as we see here, with the IgG remaining elevated for a considerably longer amount of time, this is really where we start thinking about some protective immunity with that, right? So how do we get it? There are multiple ways that we can get immunity, all right? We can break it into natural and artificially acquired, and we can also break it into passive and active types of immunity, all right? So 4 different ways that we may potentially get some immunity, all right? So initially we'll do passive. With the passive, for naturally acquired, we can think about this passive response like immunity that's conferred to that newborn either before it hatches out with the IgG that crosses the placenta, conferring a bit of immunity. That's passive and it's natural. Mother is giving it to that child. And we also think about antibodies within the breast milk, IgG and IgA. And with those secretory antibodies, the IgA molecules are super important while that child is breastfeeding. Getting these antibodies get into the gut and help to provide some immunity initially. All right. So passive, naturally acquired. Breast milk crossing the placenta. All right. Another passive mechanism that is artificially acquired would be plasma donation. When you donate your plasma, they take the antibodies and there's ultimately separation. We can do all sorts of techniques to purify those antibodies. And now you've got vials of antibody against specific things. Individuals that have a weakened immune response, if they've got a lack of ability to combat something, they're in bad shape. And in turn, may require a transfusion of immunoglobulin to help them combat whatever it is. So this artificially acquired passive immunity is picking up somebody else's antibodies that have been produced. This doesn't confer immunity. We're not giving them any cells. All they're getting are these proteins. So they get a transfusion of these proteins. It does its job and they ultimately get degraded. They don't stick around, all right? So they may and likely probably do need multiple transfusions with that, all right? And I know there's all sorts of details with plasma donation. If you've got high titers with specific things, you potentially get some bonuses, all right? I had a friend that had numerous tetanus shots when he was a kid, riding motorcycles and getting hurt. Tetanus shot, another tetanus shot. As he gets to college, starving college students need to make some quick cash, donate some plasma, right? He had a super high tetanus titer. And in turn got paid bonus money because of his high tetanus titer. And they could take that tetanus antibody and utilize it in these ideas. All right. Crazy. Any plasma donors out there? Not yet, but you're like, money? How much? Right? I don't know how much, but I think it's good. All right. Lastly, with this, the active immunity. This is immunity that's conferred just by flat out living, right? You get some infection and the immune response is stimulated and you produce antibodies. So illness and recovery. This is active, naturally acquired. Your body's doing it itself. And the active artificial is a vaccination. So not getting the infection, but getting a fragment of that that stimulates the body to say, wait a minute, this is a bad guy, and we produce antibodies against it. So either dealing with the actual thing or a fragment of it is an active type, either natural or artificially conferred. And I will quit with that. Farewell and good day.